same events that are described here. So I'm going to skip right over this. Now the synaptic delay. So again, this is going to take some time. So the time that it takes between the action potential arriving at the axon terminal of the postsynaptic, of the presynaptic neuron, and the time that it takes to result in a graded potential on the postsynaptic neuron, there's a 0.5 to 5 millisecond delay, right? So this is going to take a little bit more time, which is what we described. What is causing that delay is the change in the calcium, the time it takes for calcium to enter the axon terminal, the time it takes for the vesicles to move closer to the end of the axon terminal for exocytosis to occur. That is what takes that 5.5 to 5 millisecond delay. All right. It's not at all related to the diffusion of the neurotransmitter across the cleft. So we really want to make a distinction here of what takes the delay. The delay is really calcium entering the axon terminal, the vesicles in the axon terminal getting to dock at the axon terminal itself, and then dumping of that into the cleft. That is what takes the delay, and not the diffusion of the neurotransmitter across the synaptic cleft. All right. Let's begin our third learning objective. So compare the fast versus slow response at the synapse. And again, I talked about this in that illustration. So when the neurotransmitter binds to a receptor on the postsynaptic cell, that can generate two different types of responses. And so we're going to talk about the mechanism that results in the fast versus slow response. So we have channel-linked receptors that are called ionotropic receptors. And so one example of the receptor that the neurotransmitter can bind to is called an ionotropic receptor. These are going to be ligand-gated channels. And think back to what we said in terms of where on the neuron we have different types of receptors. Remember, we said that ligand-gated channels are always going to be found on the dendrites and the soma. So the channels that are present here can bind to something, right? They can bind some type of ligand. So that makes perfect sense because the neurotransmitter that's being released will act as a ligand and it will bind to one of these types of receptors. Now, a fast change in the membrane potential occurs when the neurotransmitter binds, and that immediately results in some type of ionic movement. So when the neurotransmitter binds to this ionotropic receptor, also referred to as a fast receptor, that automatically opens up a channel because the receptor is also acting as a channel. So when that ligand, i.e. neurotransmitter, binds here, automatically we open up that channel, ions can move. And in terms of the ionic movement, sodium is going to move more so into the cell, again, according to its ECDF, and that is going to result in the graded potential on the soma of the postsynaptic neuron. And so this is an example of a fast response. As we can see, this is immediate, simply because the receptor is also behaving like a channel. So as soon as that ligand binds, we open up that channel, ions can begin to flow, and we can result in depolarization, which is what causes that graded potential that can happen spontaneously. Now, if we think about the alternative type of synapse or the alternative type of signal, that would be the slow-acting signal. And what happens here is there are a series of steps in the way that has to happen after the neurotransmitter binds to this slow type of receptor. So the receptor here is not acting as a channel in the way that we saw with the ionotropic receptors. These receptors are referred to as metabotropic receptors, sometimes called slow receptors. These are coupled to a G protein, and the G protein is, the G protein is going to stimulate a cascade of events that will ultimately result in the signal being converted to some type of cellular response on this cell. So it's not automatic, it's not spontaneous the way that it was for the fast receptor, the ionotropic receptor. So here, the neurotransmitter binds to the metabotropic receptor, the alpha subunit on the G protein dissociates off, It travels downstream in the membrane. It's going to bind to an enzyme. This enzyme is referred to as an amplifier enzyme because the binding of this alpha subunit to this enzyme is going to trigger the generation of some second messenger. The second messenger, which is typically calcium, but it can also be other things. We'll talk about specific examples coming up. That second messenger will then result in the opening up of a channel, and then we will have ionic movement, and then will result in a cellular response. So notice how there's a lot of steps in the wave. So this response becomes very slow, becomes very delayed, because the neurotransmitter has to bind, then the subunit of the G protein receptor has to come off, and then it has to activate an enzyme, and then it has to result in the generation of a second messenger, and then we can result in either ionic movement or a cellular response by another mechanism. But this is going to take a series of steps before the cellular response can be brought about. The other option that doesn't include a second messenger is if that subunit immediately opens up an ion channel, and then we have ionic movement, and then we generate that cellular response. So even though there are a fewer number of steps in the second option, there still is a series of events that has to take place. This is not going to be a fast or immediate type of response, simply because we have to have other players get involved in order to generate the cellular response. Right? And this is referred to as signal transduction, or sometimes referred to as the signal transduction cascade, because it really is a cascade of events. One thing leads to another in order for that signal to be transmitted from one structure to the other. All right, so when the ligand-gated ion channel opens up, the membrane potential will change, and that will be dependent upon exactly what type of channel opens up. And we know this because if a sodium or calcium channel opens up, that results in a depolarization, because sodium or calcium have an ECDF that moves into the cell. So if I open up a sodium channel, sodium rushes in, that results in a depolarization. That is also referred to as an excitatory postsynaptic potential. So whenever we have the neurotransmitter resulting in a depolarization on the postsynaptic neuron or cell, that is referred to as an excitatory postsynaptic potential. And that is specifically brought about whenever sodium channels open up or whenever calcium channels open up. Alternatively, if we open up potassium channels or chloride channels, that will result in a hyperpolarization, and that can be referred to as an inhibitory postsynaptic potential. So if we think about potassium's ECDF, potassium wants to leave the cell. So if I open up a potassium channel, more of that potassium leaves the cell, and the cell ends up in a hyperpolarization state, right? So that becomes more negative on the inside. Likewise, if I open up a chloride channel, chloride is a negatively charged ion. So when chloride enters the cell, that's going to result in a negative change of the membrane potential, which can also be classified as hyperpolarization. And this is ultimately referred to as an inhibitory postsynaptic potential. So when we talk about the EPSP or the IPSP, it is the type of signal that is resulted on the postsynaptic cell, and that will be determined by specifically what type of ion channel was opened up based upon the ligand binding to the receptor on the postsynaptic membrane. Okay, so kind of connecting what we said about graded potentials earlier on in the chapter, right? Earlier on in chapter 7, and connecting that to what type of channel actually opens up to result in either a positive or negative graded potential. Now, when we have a EPSP or an IPSP on the postsynaptic cell, that will typically be stimulated by a different type of neurotransmitter. So EPSPs are typically brought about by the neurotransmitter glutamate, whereas IPSPs are typically brought about by the neurotransmitter GABA, all right? So the different type of neurotransmitter that binds to our different type of ligand will also help to determine, one, what type of channel opens up, and two, whether that ultimately results in an EPSP or an IPSP. All right. So going on, to sort of further describe this, excitatory changes make it more easy to fire an action potential, right? So we talked about that when weighted potentials are strong enough and they get close to -55, that makes it more favorable to begin an action potential. Whereas inhibitory changes in the postsynaptic membrane, or IPSPs, are making it harder to fire an action potential. So hyperpolarization is going to occur, and so it's going to take us away from that set point, and so it's going to be more difficult. It's going to require a bigger weighted potential in order to result in an action potential. The other thing that inhibitory postsynaptic potentials can do is they can act as a stabilizer for the membrane potential, particularly with the movement of chloride. So not only can they make it more difficult to fire an action potential, but if the membrane is overexcited, they can also act as a stabilizer to help bring that membrane potential back to a resting state. All right. So just comparing the fast response here versus the slow response here, and kind of what that looks like with different ions moving. So the fast response is always going to result in the neurotransmitter or ligand binding to this ionotropic receptor. And ionotropic means that the receptor is also behaving as the ion channel. So when the ligand binds and the channel opens up, sodium and potassium are both moving through this channel. More sodium is coming in, right? Remember, sodium has a stronger ECDF, so sodium is rushing in, whereas potassium is trickling out. And so that ultimately results in depolarization, and that quickly generates a fast response on the postsynaptic cell. Alternatively, if you look at the slow response, the neurotransmitter has to bind to our slow receptor. This is coupled to a G protein, which has 3 different subunits. The alpha subunit comes off, activates our amplifier enzyme. This can vary depending on exactly what tissue or what postsynaptic cell we're looking at, but an example would be adenylyl cyclase. Adenylyl cyclase is what helps to produce cyclic AMP, which is acting as our second messenger. And then that cyclic AMP can go on to result in some type of cellular response. Again, very dependent upon exactly where this tissue is, that will result in a different type of response. The other alternative is that the alpha subunit can directly activate a channel, or the second messenger can also activate the channel. Ultimately, we can have a channel opening up, which allows for ionic movement. And depending on what type of ion is moving, that will determine whether an IPSP or an EPSP is generated. All right. So if we think about a chloride or potassium channel opening up, that will mean potassium will move out of the cell, which will be an IPSP. If chloride channels open up, chloride can move into the cell, which would be an IPSP, but chloride can also help to stabilize the membrane potential, right? So chloride can have 2 different functions. So potassium moves out, that will always generate an IPSP. But if chloride moves, chloride will move into the cell, which can be an IPSP, or if the cell is overexcited, chloride can also just help to bring it back to rest or stabilize the membrane potential. All right. So if we think about IPSPs here, they are graded potentials. They have the ability to sum, as we described earlier, depending on how many of these IPSPs or EPSPs are coming in. Remember, we have a soma, and a soma can receive several different synapses, right? So if we have an abundance of IPSPs happening and a few EPSPs, those will ultimately be summed if they're close enough in space or time, right, depending on whether it's spatial or temporal summation, and then we can determine what ultimately happens in terms of whether or not action potential is generated on the subsequent neuron or cell. So the higher frequency of action potentials means that more neurotransmitters released. More neurotransmitter means that more of that can bind to receptors on the adjacent cell. And then if we have an increase or decrease in ion permeability, that can result in either more or less ion movement, which can result in hyperpolarization.